All right. Hello, everyone. We're going to go ahead and get started. Um, I hope everyone is doing okay despite the inclement weather. I hope everyone, again, is doing okay. My name is Dr. James Mungeon. I am the program coordinator for the NLTP program, which stands for the National Bio and Agro-Defense Facility Laboratory and Training Program. And today is a very, very special day. The 2024 cohort of NLTP will be presenting their virtual oral presentations. But before we kind of begin, we want to just give you an overview of the agenda for today. So first, I'm just going to give a welcoming and opening remarks, and then that is going to be followed by the oral presentation session, and then last but not least, we'll have some closing remarks, and then we will conclude our session for today. Now, before we begin, I kind of want to tell you all, just kind of give you all a snapshot of the hard work that the cohort has done for the past 8 weeks. So for those who are probably unfamiliar with the program, NLTP is a part of the NLTP program. In the summer, there's an 8-week training program where they learn standard molecular biology techniques that is related to molecular diagnostics. And basically, they do a lot of training in that regard. And also, they do a lot of training as relates to technical skills with animal handling and restraint. And then after that, all of that, it finally culminates into them writing a final report where basically they choose a particular pathogen that has been the topic of interest by NBAF. And they pretty much are studying everything from the etiology to pathogenesis to differential diagnosis and everything. And like I said before, the cohort from this particular cohort has worked very hard with making sure they have make sure everything they've written in their laboratory notebooks, make sure everything that was in there was written well. And also, when it came to presentations, I know it was very difficult, but they have worked very hard. And so, like I said, today will be a special day where they will be pretty much sharing the fruits of their labor. Okay. And so, yeah, this here is just showing some pictures of what happened throughout the 8 weeks. So as you can see here, they were able to work with all different types of large animals and small animals. They worked with goats, they worked with cattle, they've also worked with swan species as well. And then also you can see some pictures of the cohort being in the laboratory. And then also one of the highlights with the NLTP program is that we were able to visit some diagnostic laboratories. So one of the laboratories that we visited was the Alabama State Diagnostic Laboratory in Auburn University. And we also visited the CDC, where basically we were able to get a tour to the BSL-3 and BSL-4 laboratories. And again, it was a really enriching experience for everyone. And again, I just want to let them have the opportunity to kind of showcase what they have done so far. And so without further ado, I want to go ahead and start the oral presentations. And so first one will be Corey Smith and Maya Magaha. Corey Smith is a recent alum of Tuskegee University, originally from Chicago, Illinois. She will be starting her Master's of Animal Science program here at Tuskegee University in the fall. Following her completion of her Master's, her near future plans are to earn a DVM, a Doctorate of Veterinary Medicine, and practice small animal medicine with a specialty in orthopedic surgery. And then next we have Maya Magaha. Maya Magaha is a junior animal science and veterinary science major from Chicago, Illinois. She plans to enroll in the Tuskegee University College of Veterinary Medicine in the fall of 2025. And following the graduation plans, she wishes to open an animal laboratory practice. And her specialty will be theriogenology and neonatal care. And so without further ado, I'm going to go ahead and give the floor to Corey and Maya.
All right, good morning, everyone. My name is Corey Smith and my colleague Maya Magaha will be presenting on the Crimean Congo hemorrhagic fever. So today we'll be covering a quick background of CCHFV as well as the reports of the goals of our investigation, the steps that we took to complete our investigation, and then we will review the systematic review of CCHFV and the molecular diagnostic results. So just a quick general overview, CCHV was first identified in the Crimean Peninsula between 1944 and 1945, and was then isolated as its own virus in 1956 in the Democratic Republic of the Congo. It can now be found in Africa, the Middle East, and Eurasia, and it's the most prevalent tick-borne disease worldwide. So our aims of the investigation were to perform a systematic review to summarize current findings of CCHF in animals, as well as to perform standard molecular biological techniques for pathogen detection and identification. So how did we do it? For the systematic review, we utilized databases PubMed and Google Scholar and keywords such as livestock etiology and pathogenesis. We excluded articles that were written pre-2014, and we only focused on clinical trials, journals, reviews, and systematic reviews. For our personal selection, we selected 3 clinical trials, 18 journals, 79 reviews, 25 systematic reviews, leaving us with 125 total articles for review. And now I will pass it off to my colleague to review our results for the systematic review.
So what we found was that CCHFV is a spherically shaped, single-stranded, negative-sense, tri-segmented enveloped RNA virus. It is of the genus orthoneurovirus in the Nairoviridae family in the Bunявiridae order. Its number 1 vector and reservoir is Dioxidase or hard-body tick, specifically the Hyaloma species, which is also found in the endemic areas. This photo shows the life and transmission cycle of the tick, as well as its maintenance of the virus via transstadial, transovarial, and venereal means. This slide shows viral entry to host cells via the protein nucleolin, which is present in and on the surface of eukaryotic cells and serves as the attachment mechanism for viral envelope proteins. Also shown here are the target cells, which include monocytes, macrophages, and dendritic cells, which are involved in the adaptive immune response. Hepatocytes are also shown here, as well as Kupffer cells, and they are cells of the liver that are especially important as they aid in detoxification and protein synthesis and work together to maintain homeostasis. Finally shown are renal epithelial cells, which help to regulate kidney function and maintain homeostasis. Therefore, damage to these cells leads to organ dysfunction and ultimately failure when viral replication persists. This slide highlights 3 diseases often mistaken for CCHFV due to overlapping flu-like symptoms: hemorrhagic signs, gastrointestinal issues, spontaneous abortions, lethargy, and inappetence. And they include Rift Valley fever virus, Nairobi sheep disease, and Q fever.
All right, there we go. So how do we know for sure that we're dealing with a CCHFV infection, especially because animals are asymptomatic for this disease? So our research found that the 3 most used diagnostic tools are the enzyme-linked immunosorbent assay, or ELISA, immunohistochemistry, and polymerase chain reaction, specifically reverse transcriptase chain reaction. So for the ELISA, we would be utilizing cell culture or brain tissue, and the diagnostic marker is the nucleoproteins. For the immunohistochemistry, we will be looking at the antigens, and for the RT-PCR, we will be specifically looking at the viral load of the S genome. So what are the best treatment methods? From our research, we found that ribavirin and favipiravir were the best antiviral options. These stop the replication of the virus, and then in conjunction with that, we have supportive therapies. For symptoms such as fever, we have antipyretics, with the most common being aspirin, which I have pictured here. So how long until the bleeding starts? The general prognosis that I found was the pre-hemorrhagic phase, which is 7 to 9 days post-exposure to the tick. The hemorrhagic phase, 1 to 3 days post-exposure, post the initial onset of symptoms, and then the convalescence period, which is up to 12 months post-exposure. So in the pre-hemorrhagic phase, we can expect fever, headache, nausea, vomiting. The hemorrhagic phase, we have spontaneous abortion, multi-organ hemorrhaging, and shock disseminated intravascular coagulation. And then for the convalescence period, we can expect hair loss, memory loss, and fatigue, leaving us with a general grim outlook, considering that the clinical fatality rates were between 10 and 50%, with an overall mortality rate of 5%. And now we'll be moving on to the molecular diagnostics. So what we did some nucleic acid detection and characterization as well as protein detection and characterization. For nucleic acid, we did the polymerase chain reaction, and then for protein detection, we did a Western blot analysis and ELISA. Additionally, we did isolation and identification of pathogenic and non-pathogenic Listeria species in bovine feces, along with gram staining and bacteria culturing.
This slide presents in the top left RNA isolation and purification results. Our 260 to 280 ratio was of concern, and our numbers were 2.06. And synthesized cDNA, which is in the bottom left results, our purity ratio being 1.82, both within reference ranges indicating pure samples. These steps were essential for RT-PCR, shown on the right. In the amplification plot, the negative control is in purple and our samples are in orange, indicating beta-actin presence and amplification in our extracted samples. This slide shows results of the Western blot analysis on the left, results of the spectrophotometer reading indicating an intensive amount or an extensive amount of protein present, which is to be expected when you're working with crude proteins. And then on the right is the results for the gel, which indicate the presence of beta-actin within our samples at 42 kilodaltons.
Yes, I was checking my cells.
This slide shows the results of the ELISA, where here colored in green indicates positivity of samples for influenza A proteins, while negatives are shown in white. This sample shows our results for isolation and identification of Listeria species using a chromogenic and specific agar. So the rapid L mono is shown in red. The Oxford agar is the one shown turning into black, and the chromogenic agar is shown in the pale color. Here we see our results for our gram staining. On the left is a positive or gram-positive streaking for Listeria species, and on the left, on the right, we see a microscopic view of gram-negative E. coli. So what does all of this mean? What we found was that more research needs to be conducted on our animal models. We need to have studies related more securely to viral isolation and identification, as they will help us determine the mechanism of viral action and help us develop protocols, diagnostics, and treatments for CCHFV. We also need to develop new antiviral medications as well as vaccines for this disease, and that our molecular diagnostic techniques were performed. Yes, that's it.
I would like to send a great thank you to Tuskegee University's College of Agriculture, Environmental, and Nutrition Science, specifically Dr. James Mungin, who was our mentor throughout this project, along with Dr. Olga Bolden-Tiller, Ariel Worthy, and Karis Jones, along with Travis Burrell, Dr. Wong, Dr. Faraj, Dr. Platt, and of course the USDA and NBAF. Thank you. Are there any questions?
We have the Japanese encephalitis virus group. We have Gloria Scott, Tania Birdsall-Moses, and Layla Spinner. So Gloria Scott is a recent graduate of Tuskegee University from Little Rock, Arkansas. This fall, she plans to apply to grad school with the intention of pursuing a master's in animal science. Her future plans include earning a Doctor of Veterinary Medicine degree and practicing small animal medicine. Next, we have Layla Spinner. Layla Spinner is a recent graduate of Florida A&M University, where she majored in biology pre-medicine. This upcoming fall, she will be attending University of South Florida, where she'll be obtaining her master's in medical sciences. As for her future careers, she hopes to one day own her own medical practice, specializing in plastic surgery and aesthetic medicine. And then we also have Tania Birdsall. She is from Florence, South Carolina, and a senior animal science major from Tuskegee University. Unfortunately, Tania was unable to be here, but of course, she will be missed. And yeah. And so without further ado, I want to go ahead and give the floor to Gloria and Layla.
Good morning, everyone. My name is Gloria Scott, and I will be presenting Beyond the Bite, a systematic review on Japanese encephalitis virus and animal hosts with my lovely partner, Layla Spinner.
So for our agenda today, we're starting with our introduction, followed by our objectives, more specifically, specific aim 1 and 2, followed by our methods, results, conclusions, and acknowledgements.
So for an introduction of the virus, Japanese encephalitis virus is a vector-borne disease, which means that it starts as an infection and it's transmitted through arthropods such as ticks, but in this case, it is mosquitoes. So the virus has many vertebrate hosts, including pigs, horses, birds. There is a vaccine for the virus, but treatment for the disease host is very limited. So the chart on the right is showing the geographic distribution of the virus. It's showing that it is prominent in areas such as India, Sri Lanka, China, Vietnam, and so on and so forth.
So for our main goal of this virus for specific aim 1, it was to conduct an analysis of Japanese encephalitis virus, more specifically on the pathogenesis, treatment, prognosis, etiology, differential diagnosis, and diagnostic tools.
So for our methodology, we researched articles on PubMed, and we started with our virus, which is Japanese encephalitis virus, and got a total of 5,607 articles. Then we searched up certain keywords such as in animals and got a total of 3,318 articles. And then we excluded articles pre-2014 and got a total of 2,139 articles. Then next we included our 6 questions, which is differential diagnosis, pathogenesis, treatment, prognosis, etiology, diagnostic tools, and we chose 20 personal articles from each of those 6 questions and got a grand total of 120 articles.
So as for pathogenesis of Japanese encephalitis virus, it starts off with a mosquito bite entry in which pigs, who are the amplified hosts for this virus, become infected with the virus as they're bitten by infected mosquitoes. This leads to the initial replication of keratinocytes, as you can see on the picture. And then once inside the body, the virus first replicates at the site of entry, which is the skin. This leads to viremia, in which the virus enters the bloodstream, circulating throughout the animal. The virus then moves on to target specific organs, particularly the central nervous system. This leads to neuroinvasion, in which JEV can invade the central nervous system by crossing the blood-brain barrier and infecting neurons in the brain, thus causing neurovirulence and inflammation. So as for treatment options, we know there isn't a definitive cure for the virus, but there is supportive treatments. This includes phenobarbital, which is an anti-seizure management drug, azapiron, which is a sedative agent, and minocycline, which is an anti-inflammatory management drug.
So for our prognosis, we included a timeline of the virus in pigs. So for day 1 through 2, the virus enters the pig's body through the bite of an infected mosquito. Day 3 through 4, the virus spreads to the bloodstream, viremia, reaching various organs. Day 5 through 6, viremia peaks and the virus can be detected in the blood. Day 7, symptoms become more noticeable. Pigs may exhibit fever, anorexia, and other symptoms. Acute stage, day 8 through 10, symptoms can include high fever, lethargy, and reduced appetite. Acute stage, day 11 through 14, symptoms generally start to resolve. In the chronic stage, pigs typically do not enter a chronic stage with JAV. The infection is usually acute and resolves quickly. And death is very uncommon, being since the infection resolves after the acute stage. So for the etiology, the Japanese encephalitis virus is primarily transmitted by infected Culex mosquitoes. Mosquitoes can become infected when they feed on animals like pigs and birds and transmit the virus to other animals or humans through bites. Controlling mosquito populations and vaccinating animals are crucial to preventing the spread of JEB. So the picture on the right is showing the enzootic transmission of the virus. It shows how it starts with Culex mosquitoes, and then the mosquitoes transmit the virus to birds and swine. And then birds and swine transmit the virus to humans and cattle and horses. And then the virus ends with them because they are considered dead hosts, which means that the virus cannot be transmitted anymore.
So for the differential diagnosis of JEV, we first found that the West Nile virus, which has symptoms of lethargy, weakness, and lack of awareness, trembling as well. We found dengue virus, which has symptoms of fevers, rash, bleeding, and also lethargy. And lastly, we found the Murray Valley encephalitis virus, which also has symptoms of fevers, seizures, loss of coordination, and paralysis.
So for our diagnostic tools, we researched 3 specific diagnostic tools, including enzyme-linked immunosorbent assay, or as we call ELISA. It detects specific proteins, antibodies, antigens, hormones, or molecules in the sample. Next, plaque reduction neutralization test, or as we call it PRNT. It measures presence/concentration of virus neutralizing antibodies in samples, typically serum samples. And lastly, we have reverse transcriptase polymerase chain reaction, or RT-PCR, and is used to amplify specific DNA sequences.
So for our specific aim number 2, our main goal was to express the purpose of a thorough analysis of RNA quantification, cDNA quantification, analyze our PCR findings and Western blot image, and also the analysis of ELISA data.
So for our methodology, this is showing the steps we took for each process, starting with RT-PCR. Number 1, RNA isolation, cDNA synthesis, and qPCR. Next, ELISA, collect serum samples, block protein-coated wells, incubate samples into protein-coated wells, incubate with secondary antibody, add stop solution, and use spectrophotometers. Next, we have Western blot analysis, which starts with gel electrophoresis, transfers it into the membrane, blocks it, antibody incubations, and lastly, signal detection. Next, we have gram staining, starting with crystal violet stain, iodine stain, alcohol stain, and it ends with safranin stain. Lastly, we have the identification and isolation of listeria, and it starts with collecting the cattle fecal samples, next placing it in liquid medium listeria, transferring it into agar plates, incubating at 37 degrees Celsius for 48 hours, and checking for bacterial growth.
So as for our PCR results, starting with the upper left-hand corner, we have our RNA results at 966.4 nanograms per microliter. Below it, we have our cDNA results at 2,085.8 nanograms per microliter. Next to it, we have our amplification plot with the orange lines being our sample reading and the purple lines being our baseline reading.
So these are the results for our Western blot analysis. The chart on the left is showing the RNA isolation extracted from our liver samples, and we got a total of 46.27 microliters and milligrams. And next, our picture on the right is showing the other results where beta-actin is prominent and with a total of 42 kilodaltons. So for ELISA, our results, trial 1 and 4 was positive. Trial 4M1 is positive. Trial 4M2 is positive. Trial 4M3 is negative. And trial 4M4 is positive.
So during our isolation and identifying Listeria lab, we had 3 different sections per Petri dish. Our first section was rapid L monoagar, which is the red area. We have chromogenic agar, which is the lighter yellow area. And then lastly, we have our Oxford agar, which is the black area.
So for our gram staining, gram-positive E. coli sample, you can tell that it is positive due to the darker color blue. For the gram-negative Listeria sample, you can tell that it is negative due to the lightish pinkish color.
So in conclusion for our research, starting with our pathogenesis, we found that it involves the virus targeting neuronal cells in the central nervous system, leading to neuroinvasion, inflammation, and neurological symptoms. As for treatment, we found that there's no specific treatment for Japanese encephalitis virus, an amplified host, emphasizing the importance of supportive care. As for prognosis, we found that there was a high fatality in which neurological damage demands better diagnostics. As for differential diagnosis, we found that the West Nile virus, dengue virus, and Murray Valley encephalitis virus were the core of differential diagnosis. As for diagnostic tools, we found that new immunosensors and RT-PCR improved sensitivity. Lastly, as for etiology, we found that JEV spreads via Culex mosquitoes, in which the control and vaccination for the disease are essential. As for specific aims number 2, we found that our results for PCR, gram staining, ELISA, and Western blot analysis were concluded.
We would like to acknowledge Tuskegee University College of Agriculture, Environment, and Nutritional Sciences, Dr. Olga Bowden-Tiller, Dr. James Mungin, and our lab assistants, Karis Jones and Ariel Worthy. We would also like to acknowledge Tuskegee University College of Veterinary Medicine, Dr. Faraj, DVM, Tuskegee University College of Arts and Sciences, Dr. Wong, PhD, and Travis Burrow, National Bio and Agro-Defense Facility, United States Department of Agriculture, and Animal and Plant Health Inspection Services. Thank you. Any questions?
All right, so for our next group, we have Devin Angeletti, Alicia Williams, and KZ Fraser-Pringle. Devin Angeletti is a rising junior at Tuskegee University. This upcoming fall, she will be a junior at the illustrious Tuskegee University. Her aims as far as her career interests will be being a mixed practice veterinarian. And she wants to continue her career and she wants to pursue a DVM at Tuskegee University College of Veterinary Medicine in the fall of 2025. Next, we have Alicia Williams. Alicia Williams is a rising senior at Florida A&M University. This upcoming semester, she will be taking classes at Tallahassee State College in the fall. And her goal is to get accepted into vet school, where she will be specializing in surgical procedures for aquatic and exotic animals. And she hopes to apply for vet school next semester. And last but certainly not least, we have Casey Fraser-Pringle. She's from Atlanta, Georgia, and she is a rising sophomore at Tuskegee University. This upcoming semester, she will be taking her first round of chemistry and biology courses, and she plans to begin her application for TU Vet School in the fall of 2026. She plans to become a theriogenologist, a reproductive vet, and open her own practice at her own hometown of Atlanta, Georgia. So without further ado, let's welcome our Nipah virus group, and they will go ahead and present their research findings.
Hello, everyone. Like Dr. Mungin perfectly stated before, my name is Devin Angeletti. I am a junior at Tuskegee University.
My name is Casey Fraser. I am a rising sophomore at Tuskegee University.
Hi, my name is Alicia Williams, and I'm a rising senior at Florida A&M University. And today we will be presenting the Nipah virus and Nipah, bats, brains, and biohazards. And our lovely presenters are Devin Angeletti and Casey Fraser-Pringle.
So this is a little table of contents of what we'll be covering today. First, we'll start with our introduction, next study objectives, then our methods for our objectives 1 and 2, results, and conclusion. So the Nipah virus, whose scientific name is the Nipah virus, was first recognized in 1999 in Malaysia. And at first, scientists believed that it originated in swine. However, recent research has shown that it's actually grown in fruit bats, also called flying foxes, sorry. So in the original outbreak in Shanghai Nipah, there were over 300 cases and about an 80% mortality rate.
So the silent killer, what is the Nipah virus? Well, the reason why we named it the silent killer is because the Nipah virus is often, unfortunately, confused with the common cold. A lot of the symptoms that the Nipah virus typically portrays are similar to the common cold, so like a cough, fever, difficulty breathing, things along those lines. So it can, really get confused with that, but it can really get confused with the common cold. And what the Nipah virus is that it is a viral RNA envelope. And basically what that means is that it attaches to its host via its lipid bilayer envelope. And it also has different proteins. So it has the F infusion protein, has the G attachment protein, and it has the M matrix protein as well. So for our specific aims, so we have two primary specific aims. The first one is performing a systematic review of our scientific articles for pathogenesis, treatment, differential diagnosis, diagnostic assay, etiology, and prognosis for the Nipah virus. And we typically use PubMed or Google Scholar. And our second specific aim was to utilize standard laboratory practices for identifying different pathogens. So for the databases, as I mentioned earlier, we did use PubMed and Google Scholar. PubMed was our primary sources. The time range we typically picked was between 2014 and 2024. And the reason why we did this is because we wanted to have a little bit more accurate and up-to-date information regarding the Nipah virus, since it has been studied since the early 1900s. So we wanted to update our findings and make sure it's to make sure that it's up to date on the latest scientific information. And so for the articles, we selected 20 articles in total. So for us, that came out to be 110. And our keywords were pathogenesis, treatment, vaccine, intermediate host, differential diagnosis, and diagnosis assay.
All right, so the roadmap to the silent killer. So transmission from this virus can go from bats to humans or with contact with infectious bats or their fluids, whether that be on food, on pigs, because the first outbreak actually was from pig farmers. So we assume that they got it from handling their pigs. The entry, they use ephron receptors that connect to first your epithelial cells to get into the lining of your skin. So that's how you get it through contact. And then it replicates that way. And then it eventually attacks your endothelial cells and your neurons. And then it'll spread through your bloodstream that way. It weakens your immune system as well by attacking your spleen. And then it causes neurological symptoms, which they connect to your neurons, which causes encephalitis or inflammation in the brain, which eventually leads to mortality. And the mortality rates range from 40 to 70% in the past 10 years. And for treatments, we have symptom management, and the symptoms range from fever, headache, vomiting, muscle pain, and cough. And it could also be sore throat and difficulty breathing. Those are the main visible symptoms. Usually the ones on the inside, you have to test for it. And the treatment so far, all we can do is manage the symptoms. And in some cases, people have used ribavirin to stop the replication of Nipah virus in your body, but you still have to pass the disease naturally like you would any other sickness. And with supportive care, because there are neurological symptoms and respiratory symptoms, sometimes they can be long lasting. So we would just provide supportive care for those people.
So for etiology, there is a specific species of fruit bats that tends to carry the Nipah virus. This specific fruit bat we think is a natural host, or what we would call a reservoir of the virus. And basically what that means is that this particular animal can carry the virus and have minimum to no symptoms at all. So it's not typically displayed. And the primary intermediate hosts, are typically swine, pigs, and bovine cows. And the reason why, particularly for these two animals species, is because it tends to, once a bat, say, has bitten one, it tends to amplify. So we believe that that's why it tends to do that. And since this is a zoonotic virus, so basically it jumps from animal to human, it can be transmitted from animal to human, and then a human that has come in contact with an infected animal can get it. And also we have learned that human to human transmission is also possible as well. So prognosis for life or death. So what prognosis is that it is a chronological phase of how the disease progresses throughout the body, whether it be human or animal. And so for the initial infection, it typically occurs through direct contact, as I stated earlier, through whether a pig or a cow, or even say if you ate like a fruit that a fruit bat may have bitten or left along its saliva. And then after that, acute symptoms typically tend to happen. So fever, headache, cough, difficulty breathing, again, symptoms that can also be confused with the common cold, and mortality. So with the mortality, in severe cases, it can turn into encephalitis, which is the swelling and inflammation of the brain. And in those severe cases, you can go into a coma as soon as 24 or 48 hours and unfortunately pass away. And those images that I have there are different pictures of the Nipah virus if you were to view under a microscope.
So my results for differential diagnosis showed 3 pathogens. First, we have the measles. So the measles and NIVs are both in the paramyxoviridae family. So they're both negative stranded viruses, RNA viruses, and they cause acute respiratory infections and also similar outbreaks, as you can see in the pictures below. The picture on the left is a Nipah virus case, and the picture on the right is a measles case. Next, we have the rabies. The rabies and the Nipah virus cause damages to the brain and nervous system, which causes confusion and or paralysis, which we can see at the patient in the middle. Finally, we have Japanese encephalitis, which is the main differential diagnosis of the Nipah virus. So in the late 1990s, Japanese encephalitis was going around. It was the main pathogen going around. And they both show similar encephalitis and similar lesions in the brain. However, Japanese encephalitis is a mosquito-borne virus. For diagnostic assays, we have 3 reliable techniques used today. First, we have a reverse transcriptase polymerase chain reaction, which can be used as rapid tests and confirms the Nipah virus during the first stages of the virus. It targets the N gene to diagnose it. Next, we have the enzyme-linked immunosorbent assay, which is called ELISA, and it uses serum from the patient's body to detect anti-Nipah antibodies such as immunoglobulin M and immunoglobulin G. Next, we have virus isolation, which can only be done in BSL-4 laboratories as it requires the scientist to actually handle the pathogen.
All right, and now we're going to go into the standard laboratory practices that we went over that are used to identify Nipah virus, which include RNA quantification, Western blot analysis, gram staining, cDNA quantification, ELISA, and we also did a cattle fecal sample testing.
Okay, first we'll start with RNA quantification and isolation. So this extracts RNA from tissue samples, and it is a main step for cDNA quantification and quantitative PCR, and it basically measures the RNA concentrations. Next, in cDNA quantifications, it is called a complementary DNA transcription from messenger RNA, and you get it from reverse transcriptase.
All right, next we have qPCR, and qPCR works with amplifying the target DNA sequence in real time and measuring the concentration of DNA. So it's used to amplify the amount so that we can get proper test results. All right, next we have Western blot analysis. Western blot analysis is used to identify proteins by separating them into a gel or on a membrane, and it's used to indicate the target or the protein target, which is the beta-actin, and the dark lines at the 42 KDA mark indicate that there is a protein detection, which is a positive detection.
Next, we have ELISA, which looks for our antibody results. So as you can see, our trial for sample 1 and 2 were positives, and our trial for 4 results were also positive.
So for microbiology, we got a chance to work with Dr. Faraj with the gram staining techniques. So what you see here is we have a cattle fecal sample that pretty much describes whether or not if we had any positive bacterial samples in our culture. And so you can see from the blue dots, we did have some positive bacteria from that. And so that's what we believe what that is. And so for gram staining, there's a picture, we have gram negative and gram positive. And we basically had 10 steps that we had to do. We had to wipe the bottom of the biofilm slide clean, and we had to clean the top of the edge of the slide for about 2 millimeters. And we had to add crystal dye and wait for 30 seconds. And basically this is a rinse, wash, or repeat. I can't go through, all the steps for the sake of time. But it's basically a rinse, wash, repeat where we dye bacteria to see whether it's gram negative or gram positive, and then we observe our findings under a microscope.
So in conclusion, there is more research that needs to be done on the Nipah virus pertaining to etiology, prognosis, and basically all the factors, especially treatment, as there are multiple vaccines that are being developed now, but there is still no certain treatment for the virus. And for differential diagnosis, there were many spreading around that time, and there is evidence that the Nipah virus may have been mistaken as another pathogen before it was recognized in 1999, primarily because Japanese encephalitis was a huge pathogen going around at that time. And in 2018, the most recent outbreak in India, it was almost mistaken for the measles. As for the laboratory techniques, out of the many that we use, some of them can be used to identify our pathogen, the Nipah virus. And this includes the reverse transcriptase polymerase chain reaction, ELISA, and Western blot. However, RT-PCR and ELISA are the main ones. Thank you for listening. Oh, first, I want to acknowledge our Tuskegee team, our amazing coordinator, Dr. Munjan and Dr. B, and our laboratory assistants, Karis Jones and Ariel Worthy. Next, our doctors at the veterinary college, Dr. Faraj and Dr. Platt. Next, we have Dr. Wong and her PhD student, Travis Birrell. And I want to thank the NBAF training program and the USDA for giving us this amazing opportunity. Thank you for listening.
Last but not least, we have our final presenters. We have Adetunji Gillum and Akinyemi Gillum. Adetunji Gillum majors in animal science and veterinary medicine and is in pursuit of a bachelor's degree and a future doctorate. His primary work focuses on being a mixed veterinarian, concentrating on exotic animals, and is currently in pursuit of internships and future jobs related to that field. His future goal is to graduate from Tuskegee University School of Vet Med, where he wants to he wants to earn his DVM. And last, we have Akinyemi Gillum from Atlanta, Georgia. He is a rising sophomore at North Carolina A&T. He is currently in pursuit of a bachelor's degree in animal science and plans to find more internships in the future, focusing on marine life. In the near future, his next steps will be to graduate from the illustrious Tuskegee University School of Veterinary Medicine, where he wants to earn his DVM. And without further ado, I will go ahead and pass the baton to the Gillum brothers.
All right, greetings everyone. My name is Akinyemi Gillum.
And greetings everyone. My name is Adetunji Gillum.
And our presentation is going to be focused on a neglected but deadly virus, the Rift Valley fever virus. So our outline of this presentation is just we're going to quickly give an overview of the virus, go over our specific aims, our methods, the results that we got, go over a conclusion, and then lastly, our acknowledgements.
So what is Rift Valley fever virus? Rift Valley fever virus is a zoonotic RNA virus with a negative stranded genome and is primarily located within the east and south side of Africa. It's classified within the Brinyavidae family, specifically belonging to the genus Fabulovirus, and is transferred via vectors, specifically mosquito, and contact the bodily fluids of an affected host. Our gap in knowledge was identifying all the ways a host can be infected internally due to Rift Valley fever virus's variety of symptoms. Our other gap in knowledge was the impact of climate change and how would that affect Rift Valley fever virus because it's only prevalent during heavy rainfall seasons. And we also wanted to understand outbreak prediction, being able to predict outbreaks due to its seasonal nature.
So our first specific aim was to go over perform a systematic review on the Rift Valley fever virus, which would include the pathogenesis, differential diagnosis, etiology, treatment, diagnostic tools, and prognosis. So the way we conducted our research, we researched using Google Scholar. So we first, of course, looked up the Rift Valley fever virus, which came up to over 70,000 articles. So we had to narrow that down by putting in our keywords, which would be the pathogenesis, treatment, prognosis. And then we specifically tried to focus on animals and livestock. And then we excluded articles prior to 2010. And then for our personal selection, we chose 20 articles for each of our keywords, resulting in 120 total. So what I found are basically three central themes for my treatment and diagnostic tools. So when I was researching the treatment, they mostly talked about ribavirin, which is a nucleoside inhibitor, which is essentially a class of antiviral medicine that helps prevent or reduce the virus replication. They also talked about supportive care and symptom management, which can go hand in hand. But when I talk about supportive care, it's more in the severe cases where they have to be provided fluids and transfusion. For diagnostic tools, they talked about reverse transcription polymerization reaction, which is very sensitive but very specific, and it generally targets the L segment of the Rift Valley virus. They talked about enzyme-linked immunosorbent assay, also known as ELISA. It is more effective for detecting past infections, so it's not really suitable for early diagnosis and for outbreaks. And they also talked about virus isolation. So this is prognosis. So basically prognosis really depends on the species and the age of that certain species. So in this case, this is what you can expect from specifically lamb. So in day one is when the virus will start to replicate, and then day two is usually when the symptoms start to show, like fevers and refusal to move and a loss of appetite. And day 3 to 4 is usually when young lamb die off, but adult sheep can still live longer. And if they do, this is when they usually go into the acute phase, would be day 6, which where they start to experience more severe symptoms like abdominal pain. And if they get into the chronic phase, when they start to they can experience hepatocellular necrosis, and the virus can also spread to other areas of the body. And the longest they recorded sheep live was a maximum of 10 days because they died due to severe organ damage.
So how is RVF transferred? So it's transferred through arthropod vectors, specifically mosquitoes, species of the Aedes and Culex family. And so the three major themes of the etiology of the virus is that it's heavily prevalent during rainfalls where it originates, and the mosquito contracts it as a larva. And once it matures, it will target livestock or directly infect human. Now, humans are also able to contract the virus as well through livestock, through contact of contaminated bodily fluids as well. So once it's within the host, Rift Valley fever targets primarily the liver, specifically the hepatocytes within the liver, and that's where it undergoes its replication and translation. And this leads to the cell's apoptosis and liver necrosis. It also targets the spleen, brain, and spine. And within the spleen, it targets the lymphocytes, which leads to immune system damage, as well as the neurons, which leads to brain swelling as well. And due to Rift Valley fever's variety of symptoms, so there are a lot of other diseases that are commonly misdiagnosed. So yellow fever, malaria, and Crimean Congo hemorrhagic fever are the three primary differential diagnoses. And this is due to the overlapping symptoms such as headaches, fevers, and muscle aches, as well as just overall flu-like symptoms. There are also some other specific symptoms, including internal bleeding and liver failure.
So our second specific aim was to perform standard laboratory techniques for the identification of pathogens. And so some of the things that we did was nucleic acid detection and protein detection. So under nucleic acid detection would be our RNA isolation and cDNA synthesis, which were the precepts to perform PCR. And for our protein detection is where we perform the Western blot analysis and ELISA. So these are our results for our RNA isolation, cDNA synthesis, and our PCR. So the top left shows our RNA isolation results. It came out to a purity of 2.12 and a yield of 1,276.2 nanograms per microliter. The bottom left shows our cDNA synthesis results, which came out to a purity of 1.80 and a yield of 1,519.8 nanograms per microliter. And the big graph on the right shows our PCR results. The yellow line is our sample readings, and the blue line is the base reading. We also did gram staining, and so the figure on the left, far left, are two slides that we had, and the two figures on the right basically shows what they look like under a microscope. These are pictures that my brother took. And so you can tell which ones are positive and which ones are negative. So if it's positive, it would be more purple, and the negative would be more red.
And so another thing we did, we also successfully identified and isolated listeria. And this was through the use of agar cultures. And we got a sample from cattle. It was cattle fecal samples, and we were able to use the rapid mono agar, Oxford agar, and chromogenic agars to identify the different listeria colonies, which are the monogenocytes and alveoli. We also performed ELISA, and our target sample was influenza, and then we used experimental serum samples of influenza. And in trial 1, only M4 was positive, while in trial 4, M1, M2, and M4 samples were all positive. We also performed Western blot analysis, which is used to detect proteins, and our target protein was beta-actin. And so we also isolated and quantified our protein, which gave us 26.5 milligrams per milliliter, which is a very high yield, as well as our purity, which was 1.11. And so on the image to the right, beta-actin was present at 42 kilodaltons.
So in conclusion, we found that RT-PCR was the best method to detect Rift Valley virus pathogens due to its high sensitivity and accuracy. So you can have a small sample but come out with a lot of results. And we found out that there is no cure for this virus, so patients can only be provided with general antiviral medicine and supportive care. And I found that young lambs typically have the highest fatality rate of 90 to 100% compared to other livestock, which usually range around 30 to 70%. We found that Rift Valley fever virus can spread internationally through infected hosts across the sea and worldwide, and it mainly targets the liver hepatocytes for replication. And lastly, we successfully performed all the molecular diagnostic techniques.
And so we would like to thank the United States Department of Agriculture, the Natural Agro and Biodefense Facility for allowing us to participate in this laboratory training program. We would also like to thank Tuskegee University College of Agriculture, Environmental, and Nutritional Sciences, and our program directors, Dr. James Mungen and Dr. Olga Bolden-Tiller. We'd also like to thank our lab assistants, Ariel Worthy and Karis Jones. And we would also like to thank the Tuskegee University School of Veterinary Medicine, specifically Dr. Rawar Faraj and Dr. Terrence Platt, as well as the College of Art Sciences, which is Travis Baruch and Dr. Wong.
Thank you for listening, and are there any questions?